Alright guys, so I'm Sharon and today I'll be talking to you all about nanoparticle MRI contrast agents. So before I get into that specifically, I'd like to just give you guys a brief overview on MRI and the difference between T1 and T2 relaxation. So as we all know, there are a lot of water molecules in our body and they all have hydrogen protons that are spinning about their axis in a random fashion in our body. And with MRI, a static magnetic field is applied, which causes the axis of precession of those protons to either align parallel or anti-parallel with the magnetic field. And essentially what happens is that the total net magnetization ends up aligning itself parallel with the magnetic field. Sorry. Hello? Okay. Yeah, if you guys have any questions, feel free to ask throughout the. So with at this moment, after the static magnetic field has been applied, it's important to note that the protons, they're not spinning in sync. They're all out of sync with each other, which is actually important when it comes to the T2 relaxation. So after that, a bunch of RF pulses are applied to the whole system, and this causes the net magnetization of the protons to be knocked down to the XY plane. And now all of the protons are actually moving in sync with each other with their net magnetization on the XY plane. So after that, the RF pulses are removed. And when this happens, two independent but simultaneous processes occur. And so they are the T1 relaxation and T2 relaxation. And there are other phrases for them as well, as you can see. And so with T1 relaxation, essentially what that is it's the time that it takes for that net magnetization that's on the XY plane to return to about 63% of its original position. And T1 relaxation or T1 weighted images usually are presented with bright images. They produce hyper-intense signals. And these are usually more, these are preferred when you want clear anatomical images of the structure that you're looking at. And T2 relaxation, as I mentioned earlier, it has to deal with the protons' interaction with each other. And so this is the time that it takes for the protons to begin spinning out of phase with each other. It's the amount of time it takes for it to reach about 37% of what it was originally. And with T2 relaxation or T2 weighted images, they produce hypo-intense signals, so the images appear darker on the MRI scans. And these would be preferred when you want to look at, when you want information about pathological, or when you want about structures, because a lot of the times abnormal fluids appear brighter in comparison to the normal tissues or normal fluids. And so that's when that would be helpful. And something else to note would be that the MRI scans are quantitatively evaluated by their T1 times, but they can also be referred to by their R values, their relaxivity values, which is basically just the inverse of their relaxation times. So the shorter the relaxation period, the higher the R1 value, R1 or R2 value, and that just means that the stronger that signal is. And a lot of the times so now we'll talk about contrast agents, which they're basically paramagnetic species, or sometimes super paramagnetic species, that help enhance the MRI scans to help with visuality more. And so the most commonly used contrast agent in the clinic now are gadolinium-based, or GBCAs. And as you can see here, the gadolinium ions are chelated with ligands, and this is because free gadolinium ions themselves are pretty toxic in vivo. They can mess with the physiological processes in the body, and so that's why they have to be chelated with ligands that help prevent the ions from actually interfering directly with the tissues. So GBCAs were first introduced in 1988, and here's an example of what it looks like. It's a T1 contrast agent, and so as you can see in these MRI scans, after administering GBCA, the meningioma that was present in this patient was much the signal was much more intense, so you could see it much better. Now, one of the issues with GBCAs, though, is that people or patients who suffer from kidney dysfunction, if they're administered GBCAs, they can wind up suffering from nephrogenic systemic fibrosis. And,
question for you.
Yes.
How did the contrast get into the brain and the meningioma?
So the contrast agents, they're usually picked up by macrophages, and usually whenever there are a lot of near the diseased areas, there's usually a lot of like inflammation and macrophages, monocytes, and cells like that. And so the contrast agents are attracted to those areas.
That's fine. Thanks. I think partly in imaging the brain, it has also something to do with these diseases induce a leakiness in the blood-brain barrier. So you'll end up with accumulation in these tissues in the brain that make it very obvious that you have this problem, but the leakiness of the blood-brain barrier is not very big, so you don't really see much of it on the brain.
Oh, okay. Got it.
Okay, very good.
So with NSF, or with patients who have kidney dysfunction, they're unable to clear the GBCAs properly from their kidneys, and they end up, the contrast agents end up spending more time in the body, and this causes, or like spending too much time in the body can lead them to them being dissociated, the gadolinium ions from being dissociated from the ligands. And so as I said earlier, the free gadolinium ions can be toxic to humans. And so one of the effects is nephrogenic systemic fibrosis. Research has also shown, or recent investigation has also shown, that GBCAs can accumulate in brain tissues and bones. And although there hasn't really been any results as to how adverse these effects could be, but current investigation is being done into that area. So because of the issues, these issues that we have with gadolinium-based contrast agents, researchers have looked into nanoparticle contrast agents because they offer more stability and biocompatibility. And one such nanoparticle are iron oxide nanoparticles. And this group has been very heavily researched just because they're very biocompatible, and there are lots of things you can do with them. You can functionalize them to be applied for a specific targeting, or to be applied for fluorescence imaging and drug delivery, alongside serving as an MRI contrast agent. And iron oxide nanoparticles, they serve as T2 contrast agents for the most part. And this is because even though they have 5 unpaired electrons that shorten the T1 time, their magnetization is also very strong. And this is mainly because instead of being paramagnetic, they're super paramagnetic. And so what that means is they essentially have much stronger magnetization properties that interfere with that phasing and dephasing property of the protons. And so because of that, they're used as T2 contrast agents. And as you can see in the image, in the MRI scans in the middle here, in this specific study, the patient was administered a pre-contrast agent that highlighted the different parts of the brain. And after applying the iron oxide nanoparticle, you can see that there was a lesion that appeared in the right part of the brain over here. And it's dark because, as I mentioned earlier, T2 contrast agents lead to hypo-intense signals. Now, going from the iron oxide, for the remainder of the presentation, I will mainly be talking about iron oxide nanoparticles just because they have been heavily researched. But I'll also cover some other nanoparticles as well. And these synthesis procedures can be applied to magnetic nanoparticles that don't involve iron oxide. Uh, so this first synthesis method that I'll be talking about is co-precipitation. And with this method, essentially what happens is two salt solutions of iron 2 ions and iron 3 ions are mixed together. And they're mixed together in an oxygen-free environment with the iron 2 and iron 3 ratio being 1 to 2. And after doing that, a base like NaOH is applied, or it's mixed with the in the mixture, and it has a pH of about 9 to 14. And after purification, this procedure yields iron oxide nanoparticles in the form of black colloidal particles. Now, so some advantages to this procedure is that the cost is low and it's a pretty mild procedure. There isn't too much going on and it can all be done in one step. And a major advantage to this is that oftentimes these nanoparticles, their surface is hydrophobic, which is not an advantage when used in vivo, because if it's hydrophobic, then they can be targeted for like opsonization, and plasma proteins can bind to them. But instead, what researchers have done, like for instance in this 2001 study, is that they have coated the surface of the iron oxide nanoparticles with a hydrophilic substance. So in this case, in the study, the nanoparticle was coated with oleate, which is a surfactant, and the researchers did this through the co-precipitation method. And they observed that they obtained the single particle magnetite iron oxide nanoparticles, and they obtained the size that they wanted, and they also observed that these particles exhibited the supermagnetic properties of iron oxide. Now, one of the disadvantages to this type of to this method is that the particles oftentimes they can appear polydispersed, and so the morphology isn't that great. And in fact, in this case, researchers observed that some of the particles that they obtained were either oval or spherical, and so that's a disadvantage to this type of synthesis method. And so another synthesis method is thermal decomposition, and in this case, organometallic precursors are lysed in the presence of a surfactant and a solvent, and this reaction is carried out at very high temperatures. And after purification, we obtain iron oxide nanoparticles. Another term for them is spion, which you can see here. And there are two ways you can do this. There's the heating up method and the hot injection method. With the heating up method, the organometallic precursor and the surfactants are mixed together in the solvent, and then that solvent, or that entire solution, is heated up slowly. And that was the case in this 2007 study, where they heated it up, and images A and D are only 20 minutes apart. And as you can see, in just that short amount of time, they observed a rapid growth of iron oxide nanoparticles, and they observed that they were monodispersed, and they had smooth surfaces. So that's one method of synthesizing these particles through the thermal decomposition process. And the other method that also involves thermal decomposition is hot injection, which I mentioned earlier. And so that is basically where the organometallic precursor is injected into a solution that already contains a solvent mixed with the surfactant. And then that's also carried out over a period of time, and that has resulted in yielding fast-growing nanoparticles as well. Now, so with this procedure, the reason that the one advantage that it has over the co-precipitation is that you have control over the morphology of the particles. However, one of the disadvantages is that when you do it, like after the first step, you acquire hydrophobic organic or hydrophobic nanoparticles. And with the co-precipitation method, we solve this by adding a surfactant or some type of hydrophilic ligand during the synthesis. But in this case, you can't do that all at once, and you have to take an additional step to modify the surface. And so after doing that, you can acquire the hydrophilic spion, but in this case, it just takes a bit longer to carry out this process. So as I mentioned earlier, the surface coating is important. If nanoparticles are hydrophobic in the body, then the body won't really react with it well. And so a lot of procedures have been done to help to kind of investigate how to coat these nanoparticles and see what works and what doesn't work. And so in this 2010 study, the researchers were able to create multiple layers on this particular nanoparticle, and it ended up showing good results in the MRI, and it showed high circulation in the body, which is something that you would want if the MRI is being taken over a long period of time. You don't want the nanoparticle to be excreted from the body too quickly. And so that's one of the perks of coating the surface, is that you increase the retention time as well as the biocompatibility, and it's more stable in vivo. And another thing to note is that if they in the body, and that wouldn't really lead to its optimal usage.
Hey, Sharon, I have a question.
Yes. Is this?
That's Fabian.
Oh, okay.
So this functionalization, did they ever talk about decreasing the tumbling?
Decreasing the tumbling?
Yeah, or effect of the tumbling?
Yeah, they talk of. I read about that when they were trying to modify this particle so that they could make it into a T1 contrast agent. So you're talking about the actual surface coating being affected by the tumbling being affected by the surface coating?
How is that? If they talk about having any effect on the molecular dynamics of this small molecule complexes. Sorry.
I'm not sure if I recall that information.
Okay. Yeah, you're good. Just don't worry about it. Thank you so much.
Mm-hmm. So the surface coating definitely does help with all of the other aspects. And something else to note is that it helps prevent the iron ions from actually leaving the particle, which is also important because if it did leave, although iron isn't necessarily toxic in the body, a high amount could lead to bad results. So, sorry. So Brian D. Ross is one researcher that has been heavily involved in this research, and he is a professor of radiology at Michigan Medical School, and he has an H index of 172, and he's done a lot of research on nanoparticles and contrast agents in general. And what he and his researchers did in this particular case is, instead of having an actual nanoparticle coated with these hydrophilic groups, they created a polyacrylamide matrix, so a PAM, which in itself is pretty stable in the body. And then they also added polyethylene glycol ligands, or polyethylene glycol ligands to it, which are PEG. And they observed that it increased the R2 values, which means it helped increase the MRI sensitivity to these contrast agents. And an important thing to note about this is that it's a little different from the others because with the matrix, you can incorporate multiple iron oxide particles or crystals in it, which helps to increase the magnetization effect it has on the protons. And so that's one way that they've used this the coding to help. So up until now, I've talked about these nanoparticles as T2 contrast agents. But there are some drawbacks to having T2 contrast agents in general because, with iron oxide, because of its super paramagnetic properties, it carries out something called the blooming effect, in which not only does it enhance the signal in the lesion or wherever it is, but it also affects its surrounding tissues. And it can make it hard to delineate between the diseased tissue versus the normal tissue. And so researchers have been looking into developing, or have been looking into modifying IONPs as T1 contrast agents. Now, the thing about these contrast agents is that the T1 contrast agent, or the T2 property of contrast agents, deal with the diffusion of protons in the area of the particle, because it deals with the magnetic fields that are like around the general area. Whereas if we're talking about T1, the T1 values of the tissue is affected by water molecules being in close contact with the paramagnetic centers of the molecules. Now, iron oxide nanoparticles can be viewed as a core structure surrounded by a shell. And the core is highly magnetized, so is the shell. But the thing with the shell is that it exhibits something called spin canting, where all of the magnetizations on the surface itself isn't, it's not properly aligned. They're all random, whereas the core aligns with the magnetic field. And so researchers have noticed that by decreasing the volume of the nanoparticle, they're decreasing the core that actually contributes to the magnetization. And by doing that, they decrease the T2 aspect of the particle, and thereby enhances the T1 aspect. So in this 2011 investigation, the researchers carried out a thermal decomposition process whereby they acquired a monodisperse array of nanoparticles that were about 3 nanometers in diameter. And after applying it in vivo, they observed that it exhibited T1, much greater T1 properties than it did T2. And so this is a feat because T1 agents are usually more used in the clinic. Now, what if we decided to combine the two properties? That would be the ultimate kind of contrast agent. And so researchers have also been looking into doing that. And in order to do so, they one study showed that they took as I mentioned earlier, the IONP is a core and a shell. So they made the core composed of a super paramagnetic compound, and they separated it from the shell with a silicon dioxide layer, and the outer shell was just composed of a paramagnetic compound. So as you can see in this experiment, they didn't use iron oxide at all in the outer layer. And this, what it does is it allows water molecules to come into close contact with the paramagnetic centers on the surface of the particle, but also be affected by the super paramagnetic activities of the inner sphere. And so as you can see here, they were able to obtain monodisperse molecules or nanoparticles that later on, when they use it in vitro and in vivo, they observe these effects and go into play. So even though iron oxide nanoparticles do have their advantages, they're biocompatible and they're stable and they can be functionalized for a variety of different applications, they do have their drawbacks. So in this study, Story and their co-authors did this study in 2012 where they observed the liver of a patient who was administered IONP as a contrast agent. and they observed him over the period of 11 months. And as you can see, compared to the baseline, the patient, the liver at 11 months did not excrete the iron oxide nanoparticle completely. And this isn't necessarily dangerous, but it can affect future radiologic scanning that this patient may have done because this contrast agent would interfere with any future scans.
I have a question here.
Yes.
Do you mean they injected him with, or the patient with, iron oxide nanoparticles repeatedly, or just initially, and then looked at the patient every 2 months?
This was just initially. They also did a separate study where they just observed patients who were administered these particles multiple times and observed that there are iron deposits in the body, but that they're not sure as to what effects that may have. But they're assuming that having high iron, it would elevate the iron levels in the body. But in this specific case, I believe they just observed him after one administration.
Okay.
I have a follow-up question. Hi, Shahrin. Do they mention how much iron nanoparticles accumulated? Like what percentage of it was cleared out?
I'm sorry, I don't remember that specific number.
And I guess I have a second question. Did they mention anything about if there's a limit before it becomes toxic?
They didn't mention that. I don't think it's because people haven't been administered with these particles too many times, but they just did observe that people who were administered with it multiple times exhibited high iron levels, but they weren't too specific about what the concentration should be.
Okay.
When it would be toxic.
Thank you.
Mm-hmm. Any other questions? So yeah, these are some drawbacks of using iron oxide nanoparticles, and that aside, there have also been investigation into other types of nanoparticles, and one such example is gadolinium nanoparticles. And even though I said that gadolinium is toxic for patients who have kidney dysfunction, GBCAs are still commonly used in the clinic for just regular administration. And in order to improve their relaxivity and sensitivity to MRI, researchers have been trying to incorporate them into nanoparticles. And like iron oxide nanoparticles, these other magnetic nanoparticles have been used for multiple functions. So in this case, they developed gadolinium oxide doped in europium ions, and this eventually led to them obtaining a particle that could be applied for both fluorescence imaging and in vivo MRI. And so basically with this procedure, they use laser ablation in liquid, which I didn't mention earlier, but this is another way to produce this type of nanoparticle. And basically what happens is the gadolinium ion, it absorbs a photon and becomes excited, and that energy is then transferred to the europium ion, which when it relaxes, it releases it emits a photon, and thereby proving it useful in fluorescence imaging. And then the gadolinium ion itself is helpful with the MRI imaging. So another type of magnetic nanoparticle are manganese-based, and the downside to these are that manganese does have a low R1 value, but that value can be improved, and these have been employed to avoid the toxicity of gadolinium ions. And so in this 2016 investigation, a novel type of nanoparticle was produced, and mesoporous manganese silicate-coated silica nanoparticle. And with this one, the shell of the particle was it was mesoporous, and so that allowed the actual manganese the actual manganese paramagnetic centers to be available to affect the water molecules that surrounded it. And not only that, but because of the but this particle has also been used to deliver drugs, and so they loaded it with docks, and the mesoporous aspect of this particle allowed the molecules to be released from the particle appropriately. And on top of that, they went a step beyond in this procedure, and they made this particle responsive to pH. And so because of that, whenever the particle was in the presence of a mild acidic environment, it would release the drug. And so as you can see to the bottom right over here, this is just showing how improved it is as an MRI agent, because the top layer shows how it enhanced the signal of a tumor in this structure over here, but at the bottom layer is just normal tissue. And so it shows how this particle can enhance the signal of MRI. And another type of manganese.
Sharon, I have a question. Slide, are they adding simply for size, so it doesn't get excrete molecules for size, or is it so it could cross the brain?
So the PEG molecules have actually a variety of applications. So one major reason they added it was to increase its stability in the body, but also that's a good point that you bring up, being able to cross the lipid bilayer, or actually the hydrophobic part of it would help cross the lipid bilayer, but the PEG molecules themselves are good for the stability in the aqueous regions of the cells. Does that help answer your question?
Oh, yeah, it does. Thank you.
So another type of manganese oxide nanoparticle is a hollow one, and so this also helps this also in this study conducted by Insoo Lee and his co-authors, they were able to come up with an manganese oxide nanoparticle that was hollow in the inside, and they were able to load it with docks, again, to use
Shahrin, sorry, I have a question. Going back to the slide 14. Yeah, I'm just kind of curious because PEG molecule itself, usually is hydrophilic, and from that figure, it is trying to get into this lipid bilayer. How does it work? Because the surface of the lipid bilayer is hydrophobic itself.
I am not completely sure about that.
Okay, I'm just wondering because the PEG molecule is hydrophilic and the surface right there in the lipid bilayer is hydrophobic. Okay, is the nanoparticle trying to get into that lipid bilayer?
Yeah, it is to get into the cell.
Okay, I'm just wondering because you have hydrophilic surface of the PEG molecule and you have hydrophobic one for the lipid bilayer. I'm just wondering how would the particle would then go into that to the cell.
I will have to read up on that again. I'm sorry.
Okay.
So actually, hi Sharon. I had a question about that too, but I'm thinking, do you think it's maybe because of endocyte? It's just endocytose, so it's actually in an endosome.
Right. Yeah.
But the picture is misleading.
So a lot of the times, the nanoparticles, they end up going through receptor-mediated endosome. So I believe that's what was happening here.
I would say so, because you've got a pH decrease, and the pH of the cytoplasm is not low. It's 7.4. So the only way you get a pH decrease is if it were endocytosed.
Right. Yeah, a lot of the times, larger particles, they end up being phagocytosed, or they end up entering like the macrophages through phagocytosis. And then if they're smaller, they go through pinocytosis. But in a lot of these experiments, the way that they enter is through like receptor-mediated endocytosis. Do you guys have any other questions?
John?
Yeah, so in this case, they developed MONs or HMONs, and the TEM revealed that they were able to acquire the structure that they wanted, and they loaded this structure up with DOCs. And after doing some confocal tests, they found out that the actual free DOCs was able to enter the nucleus, and the HMONs were found around the cytoplasm. And so this is just another method by which they were able to utilize manganese oxide nanoparticles and use it for multiple methods, not only MRI, but also for fluorescence imaging, or for drug delivery. And finally, we have, I'll be talking about contrast agents that don't necessarily focus on the metal particle of it. And so Nicole Steinmetz is a professor at UC San Diego, and she studies she does a lot of research involving drug delivery and immunotherapy, and MRI contrast agents, which is one. And so with her her group, they focus on virus-like particles, and so they've been trying to modify TMV and physalis MV, which is another virus-like particle, and they modify it with contrast agents to allow them to serve as MRI contrast agents, along as well to serve as other types of imaging agents. And so the reason that they do this is because a lot of the times nanoparticles, they tend to get excreted pretty quickly from the body, and so they decided to modify physalis MV in this case to kind of help with the stability of the body. And so with this experiment, what they did is they included fluorescence imaging into the structure of the VLP, as well as an MRI, or as well as gadolinium ions to serve as MRI contrast agent. And then on the surface, they modified it with PEG molecules, again for stability, as well as with a protein peptide. And this peptide was specific to targeting prostate cancer cells. And so with this, they created this molecule, and they observed that they applied it in vivo in mice. And so the whole reason for this was to get them to serve as both MRI contrast agents as well as fluorescence contrast. And so as you can see here, they were able to acquire both of those goals, or they were able to accomplish both of those goals with the fluorescence images and the MRI images corresponding with each other. And they observed that a lot of the modified virus-like particle was retained in the tumor, which is what they were trying to observe. So another type of contrast agent are ORCA-based, or organic radical contrast agent. And so Jeremiah Johnson, he is a he's a researcher, he's a person who's been researching this topic for a while now, and he and his team have been concerned about the toxicity that accompanies metal ions, as I mentioned countless times before. And so the reason that's why they're looking into developing these types of contrast agents. And so one particular ORCA that they were using are nitroxide radicals, and they conjugated it to a polymer. Now, nitroxide radicals, as some of you may know, have their disadvantages in vivo. They can be reduced pretty easily. But his group decided to use that to their advantage and instead create, once again, a multifunctional agent that serves in both fluorescence and magnetic resonance imaging. So nitroxide radicals, they only have one radical, which makes them poor in terms of their paramagnetic activity. And so what they did is they conjugated a bunch of them to this polymer, and studies have shown that nitroxide fluorophores have been developed, where once the nitroxide is reduced, it helps to enhance the fluorescence aspect of the agent. And so that's what they did in this case, and they administered it to a mouse, and they observed that initially it exhibited MRI. Initially, it enhanced the MRI signaling of the tissue, and then afterwards, once the nitroxides were reduced, they were able to produce a fluorescence signaling. And as you can see here, the images, although in the MRI scan, it didn't appear, or there wasn't really any MRI or any signaling in the liver, but it did appear in the fluorescence imaging, and that occurred because mice actually have a lot of ascorbate in their liver. This is not what they had expected initially in terms of human applications, but mice have a lot of ascorbate in the liver, which reduced the compound. And but then later on, when they looked in the fluorescence imaging, they found out that the particles were actually concentrated in the liver, seeing as there was a lot of fluorescent signaling in that area.
Hello, Shahrin. I have a question to the previous slide.
This slide?
Okay, yes. So the first figure on the right, is that before adding the contrast agent, or it's the first and the second figures are same?
Right here. The MRI figures or the fluorescence figures?
The MRI figures.
So this is, they're just showing a before and after.
Okay, so the right one is the before, and the second is like after injection, applying contrast agent. Okay.
This would be, yes.
Right. Yeah, that's all. Thank you.
So in conclusion, there are a variety of nanoparticle contrast agents out there, ranging from magnetic nanoparticles to VLP-based to ORCAs, and they all have their own pros and cons. You know, they're able to be biocompatible, and they can be used to specifically target certain tissues, and they can be applied for a variety of different applications. Some of the drawbacks are that it can tend to be kind of hard to get monodispersed nanoparticles, or ones that have the size that are preferred by the scientists. And so these are just some areas that are still being looked into and being further researched. Yeah. Thank you.